You guys okay to get started?
Yeah, that's good. Can I share my screen?
I'm just going to do an intro bit. Is the sound on?
Yes, cool.
Right. Hello, everyone. Welcome to BGA24. This is session 16. And joining me today are the Tree of Life Lab team, who are going to go through their wet lab protocols. So please take it away.
Brilliant. Thank you, Damon, for that. I'm just going to... Okay. So, good day, everyone. My name is Graham. I'm a senior research assistant from the Tree of Life core laboratory. And today, for the BGA24, we'll be going through our Tree of Life wet lab protocols. So this is a sort of a presentation that encompasses some videos of our protocols, some experiences on making decision-making for our DNA extractions and DNA fragmentation parts of our protocols. Yeah, so we just we'll go through the various protocols in video format. We have members of the team here with us, and so we'll take turns to talk you through what we do in the lab to get some really good quality genomes out in the Tree of Life program. So just an overview of the session today. So we'll introduce ourselves to you all. I'll then hand over to Caroline, who will talk about the role of the core lab in the Tree of Life genome engine. And then we'll start going through our different protocols from our different parts of the pipeline. So these encompass sample preparation, sample disruption, nucleic acid extractions, DNA fragmentation, and then our QC steps. And finally, we'll just give you a brief insight into how we make decisions regarding DNA extractions to decide whether we've managed to get a good DNA extraction from our protocols. So we'll start off with some introductions. As I said, my name is Graham. I'm a senior research assistant in the lab. I mainly work on the aquatic symbiosis genomics project. So I work with a lot of marine organisms. Amy?
Hi. Hi, my name's Amy Denton. I'm an advanced research assistant in the core lab, and I'm taxonomic specialist for the chordates and perform lots of high molecular weight DNA extractions and RNA extractions.
Priyanka?
Hi, I'm Priyanka. I'm also an ARA within the core lab. I mainly focus on fungi samples, DNA extractions, and some genetic sequencing.
Okay, I'm Benjamin Jackson. I'm the taxonomic specialist for plants in the core lab.
I'm Elizabeth Sinclair, and I'm the advanced research assistant for the aquatic symbiosis genomics project. So similar to Graham, I primarily work on marine samples, but I'm also starting to do extractions of some of the Tree of Life Mesozoa, Bujaria, and Echinoderm species as well.
My name is Caroline Howard. I lead the Tree of Life core lab team. And we've been working hard to generate data for genome assembly now for around 3 years. So we're here to share it all with everyone.
Great. So I'll hand over to Caroline, and she will just take you through
Super.
Our role.
Thanks, Graham. Okay, so really I'm just going to go through a brief presentation to give a bit of the context of the Tree of Life core laboratory team, which will hopefully give you some information as to all of the detail that you will then hear from all of the various members of the team. So all of the work that we do sits under various different projects that are part of the Earth Biogenome project. So there's a long list of them here. We have the Darwin Tree of Life project, and you can see that's aiming to generate reference level assemblies for 70,000 species from Britain and Ireland. We are a couple of years into that project, and we're doing well, but there's still a lot to do, it's fair to say. We have the Psyche project, which focuses on Lepidoptera, the Aquatic Symbiosis Genomic project, which Graham and Liz have both mentioned. In that project, it's particularly difficult because we're aiming to sequence both the host and any symbiont genomes that are present in the same sample at the same time and in parallel. We then have the Vertebrate Genomes project, which has been going on for quite a long time now and is hopefully going into phase 2 soon. The European Reference Genome Atlas. And our most recent project that we started is the AEGIS project, Ancient Environmental Genomics Initiative for Sustainability. So there we're looking at lots of different genomes that are all relevant for crops and the growing of plants. So whether that is the fungi that will also be present when crops are successful, or looking at different traits within plant genomes. But really, the main reason to show you all of these projects that we're involved in is to say that all of the samples for all of these projects come through our laboratory. So we will have somebody working on Psyche samples next door to a person who's working on an ASG sample in the same lab as, Ben is working on some of his plant samples. We also have another team within the Sanger Institute that handles some of the more routine work. As we develop routine systems, we're able to kind of transfer those processes over. But essentially, the Tree of Life Core Lab is the melting pot that all of these different projects come through and then are treated with whichever protocol is required so that we can aim to get those reference level genomes. Okay, so I think this slide has been shown in many different places before, and it shows the genome engine as kind of envisaged by Mark Blackster, and it's what we've set up at the Sanger Institute here. So we go all the way from sample acquisition through the data generation to assembly, and then out to free access data that's published as soon as is possible. So the Tree of Life Core Laboratory really focuses on this part of that engine. So the DNA and RNA extraction and all of the generation of the data, which we then pass on to our colleagues in the assembly team. So what have we done so far? These data are updated regularly. This is correct as of the 29th of September 2024. We have over 6,000 different species in our pipelines, with some sequencing data having been produced. And we've reached the level of data required for nearly 2,500 of those species. So they are just over 2,500 have gone through to assembly. On the right-hand side here, you can see how those species fit into the whole kind of tree of life. So we're going right the way through, we have the plants in green, the fungi in a slightly yellow color, goes all the way around. And you can see, for example, within the reds, that would be the arthropods. And you can see the red lines around the outside are the completed genomes. So that's where those genomes have been assembled and released. And the blue bars show the species that are in progress within our systems. And you can see by how equally the blue bars are spaced, that actually we're covering lots of different areas across that whole tree of life. We do, in the completion zone, we have quite a dominance for the Lepidoptera. There are many, many hundreds of those that have now completed. And they represent a great example of samples that go through our pipelines really, really well. Others are more challenging, and as you hear the rest of the presentations, you'll understand more about which samples work well and which ones are more difficult. So I think you can see here our public releases is 1,910 assemblies have been released so far. So we're looking forward to that 2,000 assembled genomes milestone, and it will be coming very soon. But very simply then, if we go back to the core lab piece of that work, at a really base level, what's required to deliver those genomes? Well, we need to select a sample to work on. We need to prepare it in the way that it that works for it. So sample homogenization, need to do the high molecular weight DNA extraction, fragmentation upstream of PacBio hi-fi sequencing, There's also some library prep upstream of high-C Illumina sequencing. And we also do RNA extractions for RNA-seq and soon some long-read RNA iso-seq. And then how each of those processes are actually achieved is through working through a suite of standardized protocols. So if we go from the top here on the left-hand side in the green, these are our various homogenization techniques. In the pink, there are various different DNA extraction methods. Fragmentation and cleanup is shown in orange, and in blue are the RNA extraction methods. And the QR code here would take you to our Protocols.io page, where all of these have been released. So we work through these processes in a regular way with a kind of routine cadence, and that enables us to push through the samples that are successful with routine pipelines, and also identify the ones that are more challenging and go back and do some more R&D on those. The QC metrics that we collect at every point are very standardized as well. So that means that after having processed thousands of samples, we can go back and do analyses at a larger scale to try and understand even more of the complexity around this work. So I won't go into too much detail here because this is what you'll hear much more from all of our colleagues. But we have 3 different homogenization methods that are all shown here in pictures, and you'll see more about those later. Here are the extraction protocols with the QC metrics and our Kingfisher when we automate some of those processes. Here is the Megaruptor and G-tube for DNA fragmentation, and again the Kingfisher there for spry. And then here are our protocols. And at this point, there's lots of information, and to understand how a different sample might take a different pipeline, I've just mocked up a few different examples here. So, for example, a moth, one of those Lepidoptera coming into our system, we'd go through the sample prep triage process, go into power mashing, automated extraction, and then through these various different fragmentation and cleanup processes. Differently for a grass sample, that might go through bead beating rather than power mashing at the homogenization section. Has a different DNA extraction process. So here's the automated plant magatract has been highlighted. But again, looking for Megaruptor fragmentation for a hi-fi library. So a bivalve sample, these can be quite challenging. Initially, we would look to cryoprep those samples and go through the automated magatract process. However, we do find that sometimes those fail, and in that case, we would switch to the manual mollusk nanobind process, which we've seen is able to kind of rescue some of those samples that fail with the magatract extraction method. Fungi samples go through bead beating and the automated plant magatract process. So the lysis there is actually more challenging than initially anticipated, and we do favour the ULI sequencing method for fungi. And then an orchid sample is a very challenging plant sample. So it would go through bead beating and the plant organic extraction method. I'm sure Ben would tell you that they don't all finish their journey there, and some of them do go into further R&D. And then quite a recent example from our aquatic symbiosis genomics project, coral samples. Actually, we've managed to work up a really good bead beating protocol for those. So they then go into ULI or long read library prep. Okay. So here I just show you a snapshot of our team, and again another link to that Protocols.io workspace. I'm sure that everything will be shared, and I'm very keen to hand over to Graham and the team. So I'll just leave this up to thank our collaborators.
Brilliant. Thank you, Caroline. This is great. So yeah, so next we'll sort of start with our our pipeline, starting with the sample prep. Sort of each member of the team will have a go at sort of walking you through what we do in the lab. The first step of that is sample preparation, and for that I will hand over to Franco. Come.
Hi everyone. I'll walk you through the sample preparation process within our lab. All the samples in our lab are stored in -70 freezers, and dissection and the weighing occurs on dry ice so that the cold chain is maintained, which is very essential for our downstream processes. The amount of tissue required and the disruption method is dependent on different taxon group. As you see, we can handle different diverse samples. So we decide that based on which taxon group and what works best for them. And now I'll play a video of the preparation process within our core lab. Yeah. So here we start off with finding the samples. So once the sample selection process is done, we move on to picking the samples from -70 freezers, and then we transfer it to cold racks, which we take it to the sample prepping area. As you can see, all the samples are in uniquely barcoded FluidX tubes, and we transfer them carefully into cold racks, which is placed on dry ice. So we make sure, like while selecting the samples, we make sure what sample and what individual goes through different processes. And once it's assigned, people pick these samples from their workflow and start working on the sample preparation process. So this is the layout we have for sample prepping. We have a specially designed cold rack, or we can have like a cold metal table, which is used for dissection process. So here they are taking the samples out, weighing them, and then based on the weight and the organism part, we decide on what downstream process it's going to go under to. So this sample is like dissected for different processes based on the requirement. For instance, with arthropods, we prefer head sample for Hi-C and the thorax for DNA extraction. And once we have dissected and we are happy with the process, we take the sample, weigh it again, make sure it is within the range. So in the next few slides, you'll see how the weight range is like optimized for different taxon groups. And once the sample dissection is done, the sample is transferred to a new FluidX tube and then placed it in a order so that it can be moved to the right storage box and it can be picked up for the downstream processes. Okay, so there is a different ways we prep samples. The initial was the sample was prepped for a POMASH disruption method. This sample is now being prepped for bead beating process. So now we transfer these stainless steel beads. So these are the beads that we currently use for standardized bead beating process for plant samples. So here the samples are being weighed, and we also maintain the beads in cold racks. So in order to make sure the entire process is maintained at cold temperature. Now the sample is being weighed. We dissect into smaller pieces to make sure it gets disrupted properly during the bead beating process. So we do optimize the bead beating steps for different taxons, as well as Liz would carry over with the entire bead beating process. She'll explain a bit more how corals are bead beated and how they differ from plants, and we change the impact level and the time it gets bead beaten, and different types of beads that we use for different taxon groups. Now the sample is being transferred into the tube where we have the stainless steel beads. From here on, it will be picked as a batch. So we can bead 48 to 96 samples in one set. So while Liz is like showing the process, she'll explain a bit more on like how many samples can be processed at one time and what are the different tubes we use it and different beads that can be used for this entire process. And yeah, So now the sample will be transferred to a storage container, and it will be picked up for downstream extraction process.
Oh, sorry, that's very
Sorry. This is a quick overview of how different taxons are disrupted, and then they are picked up, weighed for different downstream process. As you can see, the plants and fungi are beaten, and we require 60 to 70 milligram for DNA, and 60 to 70 for high C, and 15 mg for RNA. Whereas the metazoans and chordates and other species can be cryoprepped, power mashed, or beaten, and also sponges are like squeezed for their extraction process, which Liz will walk us through in the next few slides. So these are the optimized weights and input weights that we have worked through our R&D, and we have standardized within our lab. I'll explain a bit more about the different disruption method that we have within our lab. So currently we have 3 different disruption methods, that is cryoprepping, power mashing, and fast prep. So we have specialized cryo bags which we use to transfer the samples into, and then we use the cryoprepping machine to disrupt those samples and then weigh it and then send it for downstream process. Whereas with biomasher, we dissect the sample, use the specialized fluidex tubes, Appendorf tubes, and then we use the power masher to crush the samples and then in the lysis buffer and then process it for extractions. Whereas in bead beating, we have different types of beads, which we add when we add these beads to the fluidex tubes, we disrupt them and then send it for downstream processes. Now I'll quickly show you how the cryoprepping disruption method works within our labs. So the weighing of the sample is similar to every disruption method that we have within our lab. So while weighing the sample and based on the taxon group, we decide which disruption method it needs to go to. As you can see, we handle diverse samples, and these samples need to be disrupted appropriately so that they can work well during the extraction protocol. Here the sample is being weighed and will be prepped for the cryoprepping process. Once we get the required weight for the cryoprepping process, we transfer the sample into these specialized cryo bags, and we make sure we maintain the entire process on cold racks. So the setup is then combined with an adapter, which will then be adapted to the fluidex tubes from where the sample was taken. The entire setup will be now immersed in dry ice so that the cold temperature is maintained even after the transfer into the cryo bags. Now we take it to the cryoprepping area and we immerse the sample and the whole setup into liquid nitrogen. Once we are sure about the temperature is maintained throughout the bag and the tube, we leave it on the machine and start off with a lower impact. So this lower impact of the process is to disrupt the larger tissue material. Once we know the larger tissue material is disrupted appropriately, then we move on to different impact levels. So this impact level is also being optimized for different taxon groups. For instance, for this sample, we start off with 1 and then move on to 2. And later we move on to higher impacts based on the disruption it requires. When we used to do cryoprepping for plants and fungi, we used to have a higher level of impacts compared to the mollusk and chordate samples. So we dip and immerse them into liquid nitrogen every time it has an impact, just to make sure the temperature is maintained when it's taken out of liquid nitrogen. And once we are happy with the disruption process, we immerse it in liquid nitrogen again and take the samples out. So once the disruption is done, we invert the tube and then flick the cover so that all the tissue material within the cryo bag comes back to the fluidex tube and it's placed on the cold rack. So now we move back to the sample prepping area where the adapter along with the cryo bags is taken off and the sample is transferred to the weighing board and get weighed. And based on the requirement for the downstream processes, it gets transferred into a new fluidex tubes. While prepping samples for cryoprepping, we ensure we add extra tissue material, just so that we don't lose much during the transferring bit. So this gets avoided in bead beating because the tissue material stays within the tube and is disrupted within the tube. Whereas in cryoprepping, we do lose samples in the cryo bags, but we ensure we balance out while prepping the sample for this process. Once the sample transfer is done, we then move it to its required storage boxes, and then it gets extracted or sent for Hi-C, or it gets sent for iron extraction. Yeah.
Oh.
Yeah. Thank you. Now Liz will carry over with the bead beating step.
So thank you, Priyanka. So as Priyanka said, for all our plants, we bead beat them, and the beads that you will have seen in the previous video were stainless steel 3mm beads, and we had 3 of those for the plants. The video you're viewing at the moment, however, is a novel method we've come up with for bead beating corals. Corals, by their very nature, are too hard to bead beat within the fluid X tubes. So we use special polycarbonate vials that you see here. We add 4mm zirconium grinding satellite beads to these corals, so they get a very specialized type. And the sample on the weighing boat at the moment is an Acropora species. So once I pre-chill the tubes before I do this, because I want both the bead and the tubes to be nice and cold. And then I'll add the sample, which has already been weighed and prepped, into the polycarbonate vial. I will try and break the sample up into smaller pieces before bead beating, because this gives you a much better disruption and pulverization of your sample. When doing this, you can batch the samples up and then place them into the Fluid X racks, and then these racks can be placed into the liquid nitrogen to keep your samples nice and cold before the bead beating takes place. Similar for the plants. The plants can be done, however, in the Fluid X tubes, the original tubes. So you can do up to 96 of those at a time. So that allows us to really increase our throughput of the samples and sort of batch them up. I'm underneath my gloves. I have special liners, and I've also got eye protection as well, because I'm handling liquid nitrogen during this stage. So once the samples have been pre-chilled, the lid can now be taken off, and the samples can now be placed onto the fast prep with empty tubes acting as a balance on the other side. The samples are secured down with the top layer of the fast prep, and I'll make sure that it's nice and tight before proceeding. Lid closed, and I now start the fast prep with a speed of 1800 for running for 30 seconds. This is done 3 times for corals and for plants. The reason we run for 30 seconds at a time is because the samples heat up during this process, and it's really important to cool them down again before each interval. So the samples are now placed back on dry ice. The lid is put back on the box so that then they can be resubmerged within the liquid nitrogen. So once these samples have been run 3 times, for corals, you should have thorough disruption completed, but this isn't always the case given the nature of the species. So what I'm looking for within this tube now is thorough disruption and just powder remaining at the bottom of the vial. This is probably the most challenging part of the process, is getting the powderized sample out of the bottom of the tube back onto the weighing boat. Similar to what Priyanka was saying with our cryoprep, we are accounting for loss of sample during this stage. So more material than is required will be put into the tubes in the beginning stage. In the boat, you can see that the zirconium bead remains, and we have sample remaining in our boat also. This sample is then transferred back into the original fluid X tube, and then suitable for downstream processing, either for high C libel prep or for DNA extraction. If the sample has been preserved in ethanol or another preservation solution, this bead beating stage can be more challenging because it's the sample maintains the liquid and is more wet even after the bead beating process. So flash frozen dry samples are definitely the best for this protocol. Plants don't have this issue because they come flash frozen to us. Plants as well wouldn't have this extra step that you're seeing at the moment because they are able to be bead beaten within the original fluid X tubes with the stainless steel beads instead of the zirconium satellite ones. So once I'm happy that all the material has gone into the tube, I'll replace the lid and pop it back in the freezer until the downstream processing. So now I'm going to show you a squeezing method that we've been using very successfully for our sponge samples. Sponge samples proved to be extra challenging for the team. We were struggling to get high molecular weight and good yields even after power mashing or cryoprepping the samples. So we started using the squeezing technique using the L buffer, which is made up of tris, sodium chloride, water, and PBS. The sample is put into a nice clean weighing boat or petri dish ideally, and it's cut up into smaller fragments within the buffer. Some sponges will require a lot more cutting and chopping with a scalpel blade than this one. This one wanted to break up very easily into different small fragments. So using the tweezers, I'm squeezing the tissue as much as I can to break up the tissue and release the cells into the buffer. This video has been sped up, but I can spend up to 5 to 10 minutes per sponge, depending on the how difficult the tissue is to break up and fragment. Once I'm happy that the sample has been broken up and I have cells within my my lysis buffer, I will then get 2 clean Eppendorf tubes, and I will aspirate off just the L buffer solution into 2 clean Eppendorf tubes. When aspirating, I'm taking care only to try to take the L buffer if possible, and not to take the tissue with me at the same time. Once this has been done, a second round of shorter squeezing is done, just to ensure that no cells remain in the Petri dish, and that we've captured all of the lysis buffer that we can. This second stage is much faster and is just to ensure that we've got everything we need. Using the same tip, I can then take off the remainder of the lysis buffer, filling up my tubes. So once this has been done, we now spin the tubes down to a speed, so a maximum speed up to 30 seconds, with the intention of pelleting our cells at the bottom of the tube. Once this is complete, I'm now going to take off the waste off the tubes, and then try and get all of the contents from the second tube back into the first tube. Doing it this way, I can maximize the amount of the size of the pellets that I have at the bottom, rather than just using the one tube. So the waste is taken off, leaving just the pellet undisturbed at the bottom. And then similarly for the second tube, I will take off some of the waste, but I will use the remainder of my volume to disrupt the pellet at the bottom of the tube, loosening it and making sure sometimes the pellets get sort of stuck to the sides. So I'm taking off the tube and putting it back into the first tube. This tube will now be empty and I have no further use for it. So with this tube, I'm putting it back into the centrifuge, and the other tube you can see there is just a balance. So I'm just left with one tube at this stage. And again, max speed for 30 seconds. So once this is done, I will have a very nice large pellet at the bottom of my tube, hopefully. I will now switch tips. I no longer have use of the wide bores. I can use a standard bore tip because I'm just removing the waste and I'm not concerned about the integrity of the sample at this point. The waste is removed. And now the sample is refrozen until further use later as either RNA extraction, DNA extraction, or Hi-C. That's fun squeezing.
Great. Thank you very much, Liz and Priyanka. That was fantastic. So you would have seen this from Caroline's slide. So this is the sort of range of nucleic acid extraction methods that we employ in the lab. What we'll be talking to you today is the automated MAGATRACT extraction, the plant organic extraction, and then the automated Mivana extraction, which I have highlighted in red. So we were going to show the manual MAGATRACT, but we were unable to get videos in time. So we'll just have the automated MAGATRACT, the plant organic extraction, and the automated Mivana RNA extraction. So with our high molecular weight DNA extractions, we use the QIAGEN MAGATRACT kit routinely for all of our samples that come through the lab. So this is a magnetic bead extraction method with multiple wash steps that removes contaminants from our DNA. So we've split our MAGATRACT extractions into sort of roughly 2 protocols. We have our plant protocol, which includes our fungi, lichen, and protists, and then we have our non-plant samples. So I've got 2 tables up on the slide, which just shows our different lysis master mix for both of these extraction types. So for the plant lysis, we have per sample 200 microliters of 1x PBS and 150 microliters of buffer AL, which are mixed together and incubated at 65 degrees for 15 minutes at 400 RPM. Immediately prior to lysis, we add 20 microliters of proteinase K for each sample, swirl the mixture around, and then add 370 microliters of lysis buffer to the samples, give them a quick vortex, and lyse them at 55 degrees for 15 minutes. After that 15-minute period, we add 4 microliters of RNase A, and then continue lysis for a further 45 minutes. For non-plants, similar reagents, but we just want to make one large master mix, so 200 of PBS, 20 microliters of proteinase K, 4 microliters of RNase A, and 150 microliters of buffer AL. These samples are, these reagent master mix is added to our samples, and they're lysed for 2 hours. So something we haven't shown you yet, but we've talked about, is our power meshing technique for disruption. So in our next video, we will have a sort of a clip of that. So we have our sort of samples on the cold rack, maintaining the cold chain for as long as possible. Samples are added to the tube. They can be either whole tissue or cryoprepped material that we add to new Eppendorf tubes. We add 374 microliters of our lysis buffer. And in the case of power meshing, we have these little plastic pestles for each sample added to our biomasher instrument. And then the sample is disrupted within the lysis buffer in these specially made Eppendorf tubes. So they have like a rough surface on the inside, which helps us disrupt the samples in the lysis buffer. So these, it takes a bit of getting used to, depending on the sample type. Some samples can disrupt really nicely. Some insects, for instance, take a little bit longer to disrupt because of the hard carapace, but as long as you get the soft tissue disrupted, it should be fine. Samples are briefly spun down just to sort of pellet any of the leftover tissue material, and then are ready for the heat block. So in this video, we're showing you our automatic MagAttract protocol, which is done on the Kingfisher. But prior to samples being loaded into the Kingfisher plates, we lyse samples in individual Eppendorf tubes. So samples are loaded into the heat block for 25 degrees for 2 hours. So while samples are lysing, we can also fill in our Kingfisher plates with the different wash buffers. So we have 2 washes of MW1, 2 washes of PE, and then we have also 2 water rinse steps. And so we prepare those in advance. What we also prepare is the magnetic bead and binding buffer mixture. So we have at the moment 40 microliters of magnetic beads plus 280 microliters of buffer MB, which the samples are added to once they finish lysing. So obviously when filling plates, we try to use a multi-pipette to prevent any sort of ergonomic issues moving forward. So the Kingfisher works with a magnetic head, which goes into each of these plates. And we can see there's a plastic tip comb on that first plate there, which the magnetic head goes to. And then the extraction works by transferring magnetic particles, which the DNA binds to, into the different plates. There's mixing steps within each plate, which removes which the wash buffers remove impurities from, and then is finally eluted in a volume of 130 microliters of elution buffer. So after our lysis step, we remove our samples from the heat block. Again, quickly centrifuge the samples down to accumulate any lysis buffer that's accumulated on the side, and then we transfer samples into the sample plate. This has, as I mentioned, 40 microliters of magnetic beads and a binding buffer, which helps the DNA bind to the magnetic beads. Again, we have to, at the moment, we can only sort of load one sample at a time into each wall of the plate. What we also, is important that we use is wide bore tips when working with DNA, so that we don't have any shearing during the transfer of sample into plate. And so here we are just loading our Kingfisher plates into the Kingfisher. So the Kingfisher works with deep well or short well plates that are placed onto a turntable and then scanned into the instrument. And then you can see on the sort of the top right of the instrument, there's the magnetic head, which transfers magnetic particles from plate to plate. So we can take our magnetic beads into the different wash buffers to remove contaminants, and then finally into our elution plate, and where we can elute DNA in our elution buffer. So once the Kingfisher is finished running, we remove the elution plate from the instrument, which has all our hopefully high molecular weight DNA. So at this point, we cover the elution plate and leave it on the bench where we elute overnight. So we still have the magnetic beads in the plate. The DNA is eluting overnight, and then the following morning we'll remove the magnetic beads from the plate, and then we'll have our DNA, which we will then QC. Okay, Ben, I'll hand over to you for plant organic extraction.
Okay. Hi everyone, I'm Ben. As mentioned earlier, I'm the taxonomic specialist for the plants, and here we're going to describe the process for the plant organic extraction process, which is the Tree of Life's mid-throughput reserve extraction procedure for the plant species that are too recalcitrant to yield an adequate quality or quantity of high molecular weight DNA from the plant version of the protocol previously mentioned by Graham, the Megatrek protocol. Obviously, so just starting here, very similar to the Megatrek protocol, but with different reagents and slightly different methods of working. We do standard lysis at 65 degrees pre-incubated for the lysis buffer, and then 55 degrees for the lysis. The same again, so 15 minutes for the to initially lyse the cells, and then afterwards, after 15-minute period, we'll up the speed to 900 RPM and add the RNase A to deactivate any RNA that might be in the sample. So that is the end of the 1 hour lysis period, and we will now transfer them to a centrifuge and pellet any of cellular debris which can be removed. And then we'll proceed to the next step, which is protein precipitation. Obviously, plants are extremely diverse, so they need a range of changes can be made to this protocol depending on the phenotype we're working with. Stuff like orchids, for example, have various contaminants that can carry over and inhibit long-read sequencing, while stuff like violets, for example, or the Calluna genus can have a lot of viscosity-inducing agents that can co-precipitate your DNA. So different attachments can be made to this protocol, or various additives can be included. Pre-lysis washes, for example, or additives to the lysis buffer that can be used to to stop these detrimental characteristics that are often seen. But what's been shown here is the base protocol. So after centrifugation, we will then take them out and we will proceed to protein precipitation. The lysis buffer used for this protocol is an SDS-based lysis buffer with various additives, antioxidant agents, for example, to mitigate the DNA browning, the oxidative damage. And the SDS can be precipitated with potassium ions, which then co-precipitates with the complex proteins that are found in the sample, which will then, after the fact, you'll see are removed with an organic extraction, chloroform-based organic extraction. So this is, we add chloroform to the sample, which is a phase separation, which will then remove non-water soluble contaminants and any proteins that are precipitated in the dodosol sulfate acetate precipitate. We then place them on a tube rotator for 10 minutes at 25 RPM. This is just to homogenize the chloroform and the sample to make sure the phase separation works successfully. As we've seen there, there's a range of different sample types. I believe this particular batch had a couple of orchids in, a number of algae that are seen at the front. So this protocol is used for a very diverse range of taxa and plant samples for similar phenotypes you see in plants. It will then centrifuge at max speed for 5 minutes in a refrigerated centrifuge to separate the phases and remove the waste, the non-water soluble waste contaminants from the high molecular DNA. Okay, we will then remove, you should be able to see the phases here. So on the top, we have the aqueous phase, which contains the high molecular gDNA. We have an interphase, which contains a lot of the sodium dodosol sulfate and potassium ion precipitate. And then at the bottom, we have the non-water soluble or waste phase, which contains most of the cellular, any remaining cellular debris, any organelles and the likes that may have been carried over previously. This phase separation purification step is then carried out again, which has been skipped here. But then you will then proceed to the automated DNA capture and purification using a double spray method. So we have an in-house speed bead, a Ceramag speed bead solution that is used here initially, followed by a 0.45 ampere PB bead size selection. So this is used to capture the DNA from the aqueous phase that may contain residual, some residual contaminants are water soluble, perhaps some chloroform that carries over and the likes, and this is done automatically on the kingfisher. Okay, here is seeing the Ceramag speed bead solution. So yeah, this is a one-time, primarily used to capture the DNA out of the aqueous phase and remove any residual contaminants. Yep, so then we proceed to the automated extraction on the Kingfisher. So the spry beads are magnetic based, so that is how this operates. So the outcome of this protocol, highly dependent on the species and the phenotype observed, will be 1 to approximately 20 micrograms of the high priority, ultra high molecular weight gDNA, which can then be shared and be used for long read sequencing, PABI long read sequencing. So after the automated extraction, it takes about 50 minutes on the Kingfisher. We will then remove the lids from our tri-coated FluidX tubes, and we'll transfer the elution into here, which can then be stored overnight to solubilize the DNA, and then we proceed the following day to DNA QC.
Great, thank you, Ben. Okay, and now we'll hand over to Amy, who will let us know about our RNA Nirvana extraction protocol.
So yes, for our RNA extractions, we use the MagMax Nirvana Total RNA kit, and this is a bead-based kit, so that enables us to have this as an automated process on the Kingfisher. So first of all, we make off our lysis buffer. So there's a lysis buffer in the kit, and we take 1 ml of that per sample. But we also have 0.7 microliters of DDT per sample within the lysis buffer as well. So if we do lots of samples, we make up a big sort of mix of that. And then we'll take 600 microliters of that lysis buffer with DDT, and we'll add that per sample. And that's because around 40 microliters is required per 1 milligram of tissue. And roughly for each sample that we process for RNA, we have about 50 milligrams. We power mash most samples in the lysis buffer that you'll see here. If we do process plants and fungi and other samples, though, that might have been cryo-prepped, and in that case, obviously, we won't power mash. What we'll do is we'll just transfer the cryo-prepped tissue into an Eppendorf tube, add the lysis buffer, and just give a pipette mix. But for power mash samples, as you can see, we just do it similar to DNA, add the lysis buffer, and mash the tissue. And so once all the samples are prepped, we then tend to put them on ice just to keep them cool and prevent any RNA degradation. And then as we're going to have this process automated on the Kingfisher, we'll have a sample plate, and we'll take 200 microliters of each sample and put that into a well of the sample plate. Let's see. as it's RNA, we don't need to worry too much about it being high molecular weight or anything like that, so we can use normal tips for this transfer into the plate. So once we've added our 200 microliters of the lysate, on top of that, we then add 100 microliters of isopropanol onto each sample. And once this is added, we can then place the sample plate on a plate shaker where it will run for 2 minutes, and we have it set at 950 RPM just to get all that mixed together. So there, as you can see it here, and remember to seal your plate. And whilst that's running, we'll then make sure we've got things prepared. So we will then be adding a bead mix, and this will have 12 microliters of beads and 12 microliters of an enhancer per sample. So we'll tend to make that up in a big mix there, and you'll see it shortly. Here it is. And what we do is we add 20 microliters of that to each sample. So this is what our RNA will bind to. And again, we use a multi-pipette just to make it easier for us and for ergonomics. And then the plate will return to the plate shaker, the same settings, but this time for 5 minutes. And whilst your plate is shaking, you can prepare all the other plates that you need for the automated RNA extraction. So we'll need a tip cone plate. We have 2 wash solution 1 plates, which have 150 microliters of that in each. 2 wash solution 2 plates, again, which is 150 microliters. We have a DNase plate, which has 50 microliters of a DNA solution, and an elution plate, which is being loaded here, which is 50 microliters of elution. So once those are prepared, you can start loading the Kingfisher up. And we like to sort of keep the DNA plate on ice whilst we until it's ready to be loaded into the Kingfisher as well. But once you've loaded all your plates and you click run, the Kingfisher will start running, but there'll be a process that you'll see here in a bit where halfway through the Kingfisher will stop. Here we go. So yeah, halfway through the process, the Kingfisher will stop and it will prompt you to add some isopropanol and a rebinding buffer to your DNase plate. So the DNase plate, as I said, has 50 microliters within it, and this is made from a turbo DNase buffer and a DNase enzyme. And yeah, so you're just then, you have to add the 100 microliters of isopropanol and the 50 microliters of the rebinding buffer separate to each sample. So that was the rebinding buffer, and now it's the isopropanol being added. But once this has been added to the DNase plate, you can pop that back in the Kingfisher and resume the protocol, and it will continue the run for the final wash steps and the elution. And it goes back in now. And then once it's finished, you will have a plate with hopefully all your RNA in the elution plate. So we pop that on ice, and then we'll transfer the LUITs to FluidXs, just so then it's easier for storage and also knowing which sample is which. So we just transfer those to the FluidX tubes, and then we'll also, once they've been transferred, we then tend to do some QC. So we'll do qubit and nanodrop for our RNA. And then depending on the QC, our RNA will hopefully be submitted for RNA-seq, where we send 100 to 200 nanograms to be sequenced. But yeah, it's very important that once you have done the transfer, that the RNA goes back on ice, again, just so there's no degradation.
Cool. Thank you, Amy. Yeah, so following DNA extractions, we then decide whether an extraction has passed or failed, and if it has passed, whether it will go into our PacBio low input or standard library DNA fragmentation method, which is the megaraptor on the left, or if it will go into our ultra low input method, which is the Cavaris G tube on the right. So first off, we have our megaraptor. So for the megaraptor, we have these screw cap tubes and syringes, which attach to the top of the tubes, and we transfer our DNA into these tubes, and then top up any sample with EB buffer. So to run a sample on the megaraptor, all your samples should be around roughly the same volume. So we generally aim for 150 microliters. Each megaraptor can take up to 8 samples at a time. So the amount of DNA we put in is dependent on this sort of the species and their genome size. So for species with large genomes, we tend to put in more DNA, while for smaller genome size species, we can afford to put less in. So it's just a calculation to determine how much DNA goes into the screw cap tubes, and then how much EB buffer needs to be added. And then the syringes just sort of pop on to the top of the tubes. There's no sort of screwing off the syringes in. Once they're ready, we switch the megaraptor on at the back and sort of remove the cassette so that the instrument can be initialized. Then we choose our settings. So here you can set your concentration of your samples, the volume of your sample, and then how quickly you want it to run. So we have a setting of 31 for our samples, and that should give us the insert side for PacBio Hi-Fi of between 12 and 20,000 bases. Samples are then loaded into the instrument. They clip into place nice and firmly. If there are instances where you have fewer than 8 samples, you should use a balance on the instrument. So that's just an empty tube and sort of an old syringe. And so once the instrument starts, the plungers of the syringe are pulled up and down and pushed down, and that draws the DNA through a hydropore at the bottom of the syringe, shearing the DNA into shorter fragments. So the length of time for a run in the Megaruptor is roughly 25 to 40 minutes, depending on your volume and your concentration. Yeah, and then once the run is finished, we pop out the samples, take off the syringes, and expel any DNA that's still in the hydropore. So after this process, there's still potentially some DNA within the hydropore. So it's important to get out all the DNA that you can and push the plunger down so that you've kept as much DNA for the next steps. So yeah, so this part of the video just demonstrates that there. Pop off the syringe and sort of squeeze out down the plunger to get off all your DNA. Yeah, pop the lid back on, and this is now ready for a spry cleanup, which we'll show you in a couple of videos' time. Okay, so the other method of fragmentation that we have is the Covaris G tube, and I'll hand back to Liz to talk you through this method.
Hey guys, so the reason we'd use the Covaris G tube rather than the Megaruptor is when our yield is lower than 500 nanograms per microliter. So we're similar to the setup for the Megaruptor. We take the DNA, but instead of putting it inside one of the Megaruptor tubes, we put it directly into the G tube. All of these samples have previously all been normalized to the same volume as well. So they're all set at the volume of 130 microliters within each of the tubes with the red numbers on. It's really important, very similar to the Megaruptor, to keep the volumes all the same because you'll be spinning these tubes down shortly. The G tubes can take volumes from 100 as the minimum up to 150. So you have got some sort of wiggle room there depending on the volume of your sample. And the amount of DNA you're using for this protocol is 150 nanograms compared to the Megaruptor protocol where you're using 1,500 nanograms. So it's specially designed for ultra low input. Once all the samples have been filled with the same volume, they're then put inside the centrifuge. And if you have an odd number, it's really important to use a balance as well. So you've got the right number of tubes inside. So the Covaris guidelines have said that in order to get fragment sizes of between 8 and 10 kilobases, you should be spinning at 5,000 RPM for 30 seconds. So once that's secured, sorry, that's a minute, not 30 seconds. Yeah. So once that's secured, they're ready to be spun. The idea of this G tube is that you have a chamber in the top section of the tube and with a narrowing base, and then a smaller chamber in the bottom section of the tube. So what you're aiming to do is push your sample from the top chamber through the filter in the middle, the narrow part of the tube, which is referred to as a ruby by Covaris, into the smaller section, and that's when the fragmentation of your DNA is taking place. So what I'm doing at this stage is I'm checking each of the tubes to ensure that the DNA volume has moved through that chamber and is now in the lower part of the tube. Once I'm happy with that, I'll flip them over and then spin them down again at the same time and same speed. If the samples haven't moved all the way through, which does happen occasionally, especially with more viscous samples, I will then just spin them down to a maximum of 3 times to encourage that DNA to move through. So once I'm happy the DNA is now in the lower chamber of the G-tube, so it's upside down compared to its starting position, the Cavaris kit comes with this special holder where you can very gently unscrew your sample, and it should be all remaining within the lid. Sometimes the DNA also sticks to the outside of the G-tube, so it's important to check there as well so you don't lose sort of any volume. And also it can, where the screw cap of the plastic is, the DNA can also sort of stick to there. So it's important to be quite thorough when you're aspirating to make sure you don't lose any volume. The next step of this process is a spry cleanup for the DNA. So we'll use the Kingfisher of this, and I'm going to load the samples into the plates so that we can do an automated spry for these samples.
Okay, yeah, thanks, Liz. Yeah, so as Liz mentioned there, we have sort of following our fragmentation, we have a spry cleanup, and as Liz mentioned again, we can do the spry cleanup on the Kingfisher, but we also do it manually for smaller amounts of samples. So instead of showing you some more images of the Kingfisher, I'll take you through the manual process of our spry. So we have our 2 DNA tubes there, which we transfer into a 1.5 ml Eppendorf. Yeah, so the idea of the spry is to sort of clean up our DNA sample as well as select the fragment sizes that we want. So for packed by a hi-fi, we're looking at 12 to 20 kb, and for ultra low packed by, we're looking at 8 to 10 kb fragment sizes. So then for our packed by a hi-fi spry cleanup, we use a 1-to-1 ratio of beads. So for instance, if you have 100 microliters of DNA, you would add 100 microliters of your Ampure PB beads. Once you've added the beads and the DNA together, we take a wide bore pipette and sort of gently pipette mix until the sample is homogenized. And then we'll let these samples sit for 5 minutes at room temperature, just for the DNA to attach to the beads. So after they've been sitting at room temperature 5 minutes, we add them to the magnetic racks and wait for the beads to pellet on the magnetic side of the tube, and we can remove the supernatant from our samples. We generally don't get rid of our supernatant. We keep hold of it into a new Eppendorf tube in case the spry process has gone wrong, and so we don't want to throw our DNA away. So we keep hold of it until after we do our QC. So we try gently remove the supernatant without avoiding to disturb the bead pellets. And then this is followed after we move the supernatant by an 80% ethanol wash. So this is freshly prepared 80% ethanol added to the beads and then left to wash for 30 seconds. And for each sample, this process is done twice. So we have two 30-second 80% ethanol washes during the spry, the cleanup. And then after both ethanol washes, the sample is briefly spun down to accumulate any excess ethanol that's in the tube, because this might affect sequencing going forward. So we tried to remove all the ethanol we can from the beads. And so we're just left with sort of purified DNA. So post ethanol wash, we elute our DNA off the magnetic bead. So here I'm adding 55 microliters of EB buffer to each of the samples. And again, the samples are pipette mixed with a wide bore pipette tip, just to make sure the elution buffer and the beads are fully mixed together. And then the samples are put on a heat block for 15 minutes at 37 degrees. And then once they're removed, they're back onto the magnetic rack. And then once the beads are pelleted, again, they are removed. The DNA is removed from the beads and put into new Fluidex Eppendorf tubes. Again, trying to avoid pipetting up any magnetic beads when you can. Okay, so the final section of our protocols is our nucleic acid QC that we do in the lab. So for DNA, both DNA extraction and fragmented DNA, we use the high sensitivity DNA qubit kit. For our quantity of DNA, we use the nanodrop to look at purity levels, and then we look at the femtopulse to look at the DNA quality. So that's assessing for DNA extractions, how much high molecular weight DNA we have in our extraction, and then for the fragmented DNA to see that it sits within the various fragment lengths for PacBio Hi-Fi and PacBio ULI. And then for RNA, we do the broad range RNA qubit and the nanodrop. So here we have qubit and the nanodrop. So firstly, starting off with the qubit, we add in our standards. So we have standard 1 and standard 2. The qubit we have in the lab is a strip qubit, so we can do 8 samples at a time. And that means we have to put in DNA standards for each of the 8 wells that we have in the qubit machine. So here I'm adding 10 microliters of standard 1, followed by 10 microliters of standard 2. And then we add 1 microliter of our DNA to each of the sample tubes before adding the buffer and dye mixture to each of the samples. So the qubit buffer has a dye in it which attaches to the high molecular weight or the double-stranded DNA, and that will, through fluorescence, will tell us how much DNA we have in each of our, or per microliter of sample. So here is a buffer and dye mix that we have in our lab, and for each of the sample tubes, we add 199 microliters of this buffer dye mix to 1 microliter of DNA. And then for the standard tubes, we add 190 microliters of the buffer dye mix into each of the standard tubes. So first of all, we run our standards. Instrument, first standard 1. Standard 2. And then if our standards are correct, we're able to proceed to measuring the samples. Yeah, so once all the samples are run, we have a list of sort of DNA concentration levels. That's DNA nanograms per microliter. We've been able to calculate the yield of our sample by multiplying that by the volume of our DNA. The other instrument we use is the nanodrop. This, first of all, we start by blanking the instrument with the elution buffer that we've used for our DNA sample to measure the baseline. And then we can add our 1 microliter of sample to the pedestal and measure. The nanodrop also gives you concentration values as well as purity levels. So this instrument measures the absorbance level at A260. So for the nanodrop, we're working 1 sample at a time, unfortunately, compared to the qubit where we had sort of 8 samples at a time. So it's a little bit more time consuming, but it gives you good purity values. Okay, and then finally we have what we call the femto pulse. So this measures the high molecular weight, the amount of high molecular weight DNA within your sample. So first of all, we go on and we will work in these Eppendorf plates where we have 11 samples per row and plus a ladder in row 12, which we run a ladder which is 165,000 base pairs long, and so that can give you a good indication of the quality of your high molecular weight DNA. So we start by adding what's called the diluent marker to our sample, so like a buffer that we will eventually put our samples into. So So we load that into each of the sample wells. Again, using a multi-pipette to make sure we're working ergonomically. And then any wells that is not going to have any DNA sample into, we add what's called a blank solution to there. So we add 20 microliters of the blank solution. So we added 18 microliters of diluent marker, and then we add in 2 microliters of diluted DNA. So I think the femto likes a DNA concentration of 0.25 nanograms per microliter. So any of our extractions or fragmented DNA, we dilute down to that concentration before we load them into the femto. We give our dilutions a quick mix with the wide bore pipette tip, because any high molecular weight DNA might sort of settle on the bottom of the tube. So to ensure that all the DNA is in solution, we pipette mix and then take out 2 microliters of DNA and add that to the 18 microliters of diluent marker. And again, we pipette mix with the wide bore pipette tip, the sample and the diluent marker to make sure it's homogenized. So here we're only loading 2 rows, but on any given day we can load up to sort of 6 to 12 rows of samples, depending on how busy we've been in the lab. And yeah, and finally the ladder working solution is added to the end, which has our ladder, 165 kilobase ladder, and then to the top we add mineral oil to avoid evaporation of our samples while they are sitting in the instrument. So yeah, important to get rid of any air bubbles from your samples, because the femto pulse has a capillary array. So I guess if there are bubbles in there, then it's going to be problematic for getting good results from the instrument. Yeah, so we make sure that no air bubbles, and then we've got the mineral oil on top, and then we're ready to load the sample to the sample tray. So we add our sample names to the software so that we can identify which sample is in which well. And generally we specify if it's a DNA fragmented sample or a DNA extraction. It helps us with our downstream analysis of the results from this instrument. And then we generally specify if a sample is a blank sample. So that's all our lab videos for today. But before we wrap up, we'll go through some of our DNA decision making in the lab. So our pipeline has 2 decision making points. So post DNA extraction, we assess whether an extraction is acceptable to pass forward into DNA fragmentation, and through which route of DNA fragmentation we're going to take it. And then post DNA fragmentation, we assess whether the sample is of good enough quality and if we have enough DNA to proceed for sequencing. So both decision points assess both DNA yield, the quality, and also take into account the genome size of a species. Down below, I've got a great example of a beautiful high molecular weight DNA extraction, which we get every week, I would say. Generally, no. And then on the right, we have an example of a sample that's gone through our DNA fragmentation. So for the high molecular weight DNA, we're looking obviously for the size of DNA above 100 kb. And then for PacBio Hi-Fi, we're looking at fragment size of between 12 and 20 kb for standard library, and then for ultra low input library, slightly shorter, between 8 and 10 kb. So for DNA extractions, we've grouped sort of femto profiles into good and bad profiles. So obviously, the best would be just a nice pure high molecular weight band, but we get sort of variations of that. So we get slightly degraded DNA in conjunction with a high molecular weight band. We also get this, what we call a smear. We also get smears with low molecular weight bands. We get some instances where we have a high molecular weight band plus some really, really high ultra long stuff. And then we get sort of DNA that's mainly smeared, but with the bulk above 50 kb or below 50 kb. And then we have really poor profiles where there's sort of no trace at all on the femto, or where we've got this really low molecular weight band only, so under 10 kb band of DNA. But then we've got sort of combinations of the smear with a low molecular weight band included. So there's a lot of sort of profiles that we get, as you can see from this table. And so DNA extraction can go down, two routes of DNA fragmentation. So a PacBio or low input library, or the ultra low input. As Liz has mentioned, for DNA extractions, we're looking at a yield of above 500 nanograms of DNA that falls under a good profile. So something that sits around 500 nanograms, if it has a nice high molecular weight band, we would probably send it through the PacBio standard library prep. Anything less than 500 nanograms, we might hold off putting it through PacBio standard library. And then in the case where the genome size is less than 1 gig, we can send that through ultra low input if the yield is between 500 nanograms and 30 nanograms, and falls under that good profile in the table that I've showed previously. So here are a few examples of our profiles that I've mentioned. So here we have a high molecular weight band, sort of one strong peak of high molecular weight DNA. We have a gel representation on the right here. You can see the nice strong band above 165 kb. So this is almost the perfect DNA extraction for us. But then we get sort of, all different types of DNA from high molecular weight band to low molecular band. So here we have a high molecular weight band with a smear, still a very good profile. The average fragment size is sitting at about 88,000 bases. And we gain a nice strong peak in this gel image. And then we get sort of slightly poorer profiles, but still acceptable for us. So here the bulk of the DNA is sitting above 50 kb. You can see the average fragment size is 97 kb. We've lost that nice sort of strong band in this gel image here, but still a lot of good high molecular weight that we can work with for DNA fragmentation. Again, slightly poorer still. Bulk of our DNA is below 50 kb. Again, no strong band in the gel image. It's still something we'll definitely work with and consider taking forward to library preparation. And then here we've got something that sort of outright we wouldn't be able to work with. So all of our DNA is sitting under 1,300 nanograms here. You can see in this gel image, sort of a strong band sort of hovering around the 150 kb mark here. So these are sort of our 4-5 main profiles of DNA we get, but we get sort of lots of variation in between all of these profiles that we have to take into account when we're making our decisions. So here are some examples of decisions that we've made in the past. So here's an extraction from a coral, a nice, strong, high molecular weight band peak here, but then also some degraded DNA. Most of our DNA is above 50 kb. So all in all, a good looking extraction profile. The yield of DNA is 1,022 nanograms, and the genome size is 0.8 gigabases. So for us, we would move forward into PacBio standard library prep for this extraction. Here, another good looking profile, a really nice high molecular weight band peak here, some smaller stuff here, and a little bit of a hump of smaller stuff here, which is possibly RNA. So yeah, all in all, a really good looking extraction from this algae. However, the yield is only 78 nanograms. So we won't be able to put this through for the standard library PacBio. However, the genome size is 0.45 gigabases. So something that we could put through for ultra low input library. So we'd put this sample through the G-tube. Here we have a sponge, Petrosia. You can see here, really, really low. Most of our DNA is sitting in this low molecular weight region here. And there's some sort of bit of a smearing to the right here, but all in all, not looking good. However, the genome size is 9,700 nanograms. I mean, the yield, sorry, and the genome size is 0.3 gigs. So something that we would consider doing, because we have a really high amount of DNA, is using a product that PacBio produces called a short read eliminator. So this is a buffer that you can add to your DNA that will get rid of all of this short molecular weight DNA here, and then hopefully pull out, and hopefully you're able to see if you've got any higher quality DNA to the right of this low molecular weight peak here. So something we can work with because of the high yield in the sample. We then do the QC again, send it through the femtome, and then reassess to see if there's any high molecular weight DNA that we can work with. Here we have a fish. Again, another high molecular weight, a low molecular weight band to the left of the sample, but also you can see to the right we have some high molecular weight DNA that's potentially usable. Again, another high yield in the sample, so almost 11,000 nanograms. Genome size is 1.9 gigs, so unsuitable for ultra-low input, but thankfully the yield is really high. I think the sample was taken through the DNA fragmentation, so given a spry to remove this low molecular weight DNA, and then the resulting high molecular weight DNA was put through the megaruptor and spried to slightly short, I think about 11,000 kb fragment size, but a really good looking profile that was, I think, sent for sequencing, and I think we got good yield for that CCS yield, yeah. So another example is from the dolphin. So this is our DNA profile on the right here. As you can see, sort of a lot of shared DNA, but in many instances in the tree of life, we've kind of given one shot at a sample. So this, I think we got one one lung sample of the species that we had to try separate out for DNA extraction, high-C, as well as RNA sequencing, and our extraction profile came out like this, which kind of looks appropriate for what we term as a direct submission. So we have found that if our DNA falls within the fragment size, the ideal fragment size for PacBio standard library, so between 12 and 20 kb, if we do a spry cleanup beforehand, we can generally submit that DNA straight for sequencing without having to do any DNA fragmentation on the sample. So the yield was really high here, so we had a lot of DNA to play with. So we could take a subset of this DNA, do a harsh spry on this to try tighten up the peak of the DNA. So post 0.45x spry, we came up with a better looking profile, falling into the ideal fragment size distribution for PacBio Hi-Fi. And again, this was sequenced, I think, on the sequel, and it came back with really good CCS yields. So this is a method that we've employed often when we sort of are unable to get high molecular weight DNA from samples, but the fragment size of the DNA falls within the fragment range for PacBio standard library prep. Finally, we have a fungi here, again, very reminiscent of the coral, the sponge sample that I showed you earlier. A really high molecular weight peak here. I mean, in all intents and purposes, this is a failed extraction. It's just one low molecular weight band of DNA. The yield, though, was very high. So we had 17,000 nanograms, and the genome size is tiny. So all we're looking to get out is minimum 150 nanograms of DNA of something that we could use to put forward into library prep. So again, we did a 0.45 spry of this. And as you can see, after that, after getting rid of that low molecular weight band of DNA, we've come up with a really sort of promising profile here. So this is sitting within average fragment size of 38 kb. So something we could definitely work with and try fragment for either standard library or ULI library. And it's something that we did. So we were able to send this DNA through the megaruptor and produce shear DNA of 3,700 nanograms of good quality or good fragment size distribution for PacBio library prep. So yeah, it's not always doom and gloom with some of our samples. Even though it looked really poor to begin with, I think we're able to work with the sample, get rid of the low molecular weight stuff, and be able to recover something that's usable. So that's the end of our session today. Like something that Caroline had in her slides is that we're really open to feedback on our protocols, which you can find at this link here on protocols.io. We also hold monthly open R&D Zoom calls on each of our taxonomic groups. So we have plants, chordates, other metazoa, fungi, protist calls, and so these alternate on a monthly basis. And so we really encourage people who are interested to email the TOL admin team at Sanger here, and you will be added to the mailing list if you're interested in attending any of these Zoom calls. Yeah, and then a reminder that we have a follow-up session on Thursday at the same time. It's an interactive decision-making session. So we've shown you a handful of profiles here, but on Thursday, we'll show you some more profiles and give you the chance to tell us how you would, what decisions you would make with those either extractions or DNA fragmentation preps. We're also going to provide the opportunity for you to ask any tricky or difficult samples that you may have encountered. We'll have our taxonomic experts on hand to sort of give advice, answer any questions about these samples. If you have any questions that you'd like to send in advance, please do email me at the email address provided there, and we'll I'll be happy to include those in Thursday's session. But yeah, that's all from us. But if you have any questions, please do let us go ahead.
Right, well, thank you very much. We've got a couple in the Discord now, all of which can capture. Will the videos be made available? The individual methods that you showed?
I think we are planning to put them onto Protocol now. Yeah. At some point, we'll connect them somehow.
I think so. I think they'll go on YouTube from BGA.
Okay.
And yeah, we'll be keen to include those links when they're available into our protocols.io methods, so that people can look at the method and see someone doing it. Hopefully.
Cool. Can a link be put to them on the Ergo Knowledge Hub?
I would love to do that, actually. I was in a meeting, I think, with Katia recently, and we were looking at that resource in the meeting. And I immediately came from that to ask whether we can share this material. And the answer is a yes. We just need to figure out exactly how and when we do it, but for sure.
Cool. Since the setup you're showing is pretty optimized for high throughput, do you have one or several kingfishers?
Yes, we have 2 kingfishers.
Do you think this could be scaled down to smaller distributed labs not working on many genomes at a time? What could be the difficulties, if any? Quite a big question, I imagine.
Well, so I think all our processes are also, we can do manually, so they don't have to be automated on the Kingfisher. So you could definitely perform most of what we've shown you today sort of on the bench top without a robot. But in terms of sort of a smaller scale automation, without a Kingfisher, I'm not sure what sort of the options are. I think Eppendorf have a sort of a smaller automation machine, I think, which is not as big as the Kingfisher, which could do fewer samples.
It's also fair to say that we began by doing all of these processes manually. We brought in the Kingfishers only after a year or so. So the protocols are available. It is doable. It just is a different scale.
How long do some of these protocols take to develop? See, we've been working on ASG at Sanger for a while now. And I know they are full of trouble. How long has it taken to develop that kind of protocol?
I don't know. How long did it take to develop PO?
So the plant organic extraction, the initial development phase was about 4 months of trial and error. It's still an ongoing process for a lot of samples. Like I said, it depends entirely on the phenotype you see. But yeah, I think it's always an ongoing thing. But the initial phase is normally a couple of months to get a general protocol that works for most things.
Yeah, so generally for most sort of protocols that we develop, we have a panel of like R&D species. So we'll sort of develop the protocol conceptually, try them on these panel species. So for plants, I think it represents a sort of a diverse taxonomic group. And so you trial them on those, and if you get somewhere, you can try them on. If you get good results from that trial or multiple trials until you've figured out something that works, then you can sort of try on a small scale of production samples to see if it sort of translates into normal production samples and then roll it out. But yeah, I think sort of figuring out what the problem is you need to solve is the first thing, and then sort of seeing if we can develop a good experimental design to test what if we can fix that problem. Then yeah, having the right samples to try it on as well. For the sponge squeezing, we did the same thing. So we were, some people gave some advice for squeezing of the sponges, and we firstly tried it on some samples that we know have worked so with the sponges, we had some samples that worked for our normal extraction and some that didn't. But the majority weren't working. So we tried it on samples that we know did work from other extractions, and then those that didn't. And then when we found that those that didn't work on our Magatract extraction protocol or disruption protocol worked with sponge squeezing, we were able to roll it out. So that took, I would say, about a month to figure out. But that was based on a protocol that was shared with us. And so we just had to roll it out. And then I think the adjustments we've made to that have been to also use those sponge cells for high-C sequencing. So I think previously that method was only used for DNA extraction, but we've also found that squeezing sponges into a nice pellet is also proved relatively successful for getting good high-C data from sponges.
Cool. Right. Are there any other questions from attendees? Feel free to unmute and talk. That's fine too. Okay. Well, in that case, thank you all for attending, and thank all of you for giving up your time to do the session. And hopefully we'll see you all in the Busco one later today. Goodbye. Stop.